Good morning, viewers. This is Kasturi Day. Welcome to MouShauk. And today I will discuss the economic importance of bacteria and fungi, the third part, that is economic importance of fungi, along with the general studies of the fungi. Okay? So this chapter, this much is left. Okay, and for this, to start with, I will request you to please to subscribe my channel if you have not subscribed yet. And if you are a new viewer to my channel, because that will notify you as soon as a new video is uploaded. Okay. And if you are liking the video, press the like button, share with your friends so that they can also be benefited from this video. And do meditate before you start your day. So while starting our day, that is starting with the discussion, we will meditate for a short period of time, and then we will start with that discussion. Okay. So for meditation, please concentrate at the point of light. Think that light is falling on you. You're feeling energetic, powerful, and happy. And you're getting different support. Helping you in your work you're doing. You're taking interest in the work. And becoming successful at the end of the day. And at the end of the day, you're thanking God for being the day. And before the day. Thank you for joining me for the meditation. And then we'll start with the discussion now. Okay. So first we will come to the fungi, the general study. A general study of fungi we will discuss, and then we will start with the economic importance. So if we go directly to economic importance of fungi, so what does fungi will not be able to know? Okay? So first we need to study the general characteristics of a fungi. Okay. Fungi are generally not plants. Why? Because they do not have chlorophyll and their body is not differentiated into roots, stems, leaves, and flowers like the plants. Okay. But they are eukaryotes and they are multicellular. Not like bacteria. They are not prokaryotes. They have a well-defined nucleus which is bounded by a nuclear membrane, and these fungi are multicellular and not unicellular like bacteria. Okay. As they do not have chlorophyll, their mode of nutrition is heterotrophic, like bacteria. Okay. Generally, they are poisonous, but some are edible also. Most common are molds which are growing on our food, bread, fruits, then leather, barks of the trees. Okay. Example are mushrooms and toadstools which are growing in the warm and the rainy season. Okay. And then Rhizopus is an example of the mold or a fungi. It's a common bread mold which grows on the bread, fruits, animal dung, leather goods, and they grow in the warm and the humid climate. Okay. So the Rhizopus looks like this. They have a mycelium. What is mycelium? Mycelium is a network of hyphae. These are the hyphae. You can see the root-like structures or the stem-like structures which they have. Okay. One joined to another, those are known as hyphae, and a network of these form the mycelium. Okay. From these hyphae rises the sporangiophore, which bears the spores. Okay, which bears the sporangium, which in turn bears the spores. Okay, inside the sporangium lies the spores. Okay. Now the sporangium looks like this one with the sporangiophore. The stalk-like structure is sporangiophore, which has a bulb-like structure, which is the sporangium, which contains the spores. Okay. Now the hyphae are the thread-like structures. Okay. Transparent thread-like structures. They penetrate when they penetrate inside our food, they will secrete one or more enzymes. okay, secrete enzymes, certain enzymes which digest the food and in the bread substratum, and they absorb it. Okay, it makes it soluble and they absorb it. Like this, so you can see on the bread, you some black spots, no, powdery structures. So those are the rhizopus. Okay, and the entire network of these hyphae is known as, excuse me, mycelium. Okay, now this sporangium is the swollen tip of the hyphae which becomes erect. Okay, and this erect one is the sporangiophore, that is the stem stalk one is the sporangiophore, and the swollen tip is the sporangium. It reproduces both asexually and sexually, but generally asexual reproduction is there. Okay, this asexual reproduction is through sporangium when it bursts to liberate the spores, scattering by wind. Okay, and falling on a suitable substratum, the spores germinate, which when get warmth and moisture, they will form a new mycelium. Okay. The A picture is of the rhizopus, which is growing on the bread. This is the A1 picture. And this one, B, is the mature sporangium, which contains the spores. And the after bursting of the spores is the third C picture is of the sporangium, which bursts which bursts and by releasing the spores. Okay. Now we come to the yeast. Yeast is also a fungi. Okay. It is also a fungus, which they are generally separate spherical cells. Okay, all is found as separate spherical shells. Okay, cells. Then separate species of yeast have come under the genus uh, Saccharomyces. Okay, Saccharomyces, the genus of the yeast. Several species come under this genus. Okay, all are one-celled fungi. These are singular cell fungi. Okay, and they occur either singly or in the form of budding chains. They are found freely in the air. Readily grow in their main suitable substrate is the sugary solutions. All kind of sugary solutions in any plant exudations which contains sugar, that is their suitable substrate to grow. Okay. For example, grapes, nectar of flowers, sugarcane juice, etc. Their shape is ovoid, oval shape. Ovoid means oval like. Okay? So their shape is ovoid shape. They have a distinct cell wall. Okay, and they have large vacuoles. One or more vacuoles are present. They have cytoplasm, well-defined nucleus that is bounded by a nuclear membrane. They have fat vacuoles. Okay, and some granules of food are also there, and some cellular bound organelles, membrane bound organelles are also present there. So they can directly absorb simple sugars, glucose, but cane sugars or sucrose has to be first broken down into simple sugars by one of their enzymes, then they are absorbed. Okay, they respire anaerobically. And when they respire anaerobically, they produce ethanol or ethyl alcohol, and 2-ATP is produced. Anaerobic respiration of glucose. We have studied earlier also, it produces ethyl alcohol or ethanol, carbon dioxide, and 2 molecules of ATP, sorry. Okay. Now we come to the economic importance of fungi. Why fungi is important to us, economically important to us. Okay. Mainly, they are used in alcoholic fermentation. Okay. The quality of ethanol or alcohol produced for beverages varies with their material fermented and the kind of yeast employed. So main importance is that they are used for alcoholic fermentation, and the alcohol or the ethanol which is produced for the beverages Okay, beverages are tea, coffees, drinks like that, those used. So those are the substances which add energy type of thing to our body. So those are called beverages. So beverages for the alcohol or ethanol which is produced for beverages, that varies with the material which has been fermented and the yeast which has been employed for this fermentation. Okay, the if the material which is fermented is more strong and the yeast employed is also different species, then that can produce some strong ethanol, strong quality of ethanol. Okay. Now, useful and harmful fungi. Okay. So we have some useful fungi, which are several species of penicillium. These are very useful fungi because it produces what? Penicillin drug. What is that which produces penicillin drug? Penicillium chrysogenus. Genum. Okay. Chrysogenum is the uh, species, okay, which produces penicillin drug, which is used as an antibiotic by, first it was discovered by Alexander Fleming, okay, and several other species of penicillium are also used for processing flavored cheeses, then for flavoring teas, okay. Then there are certain harmful fungi are also there. Many penicillium species are also there, which are very harmful to our body. Okay, to which they harm to us. Okay, like Penicillium italicum, that is a blue mold, and aspergillus, that is green mold. Even Penicillium digitatum is also considered to be as green mold, which grows on the outer skin of overripe or rotten oranges or lemons, citrus fruits. Okay, when the citrus fruits citrus fruits, they overripe on their outer skin, a green mold grows, that is Penicillium digitatum. Okay, that spoils the food. Then useful role of the fungi. Mainly, there are two useful roles for fungi, that is in medicine and in the industry. Okay. So in medicine, we know Penicillium notatum is the first antibiotic which is produced from penicillium. Okay, first it was known as Penicillium notatum, and now it is known as Penicillium. Just a second. It is chrysogenum. Okay, now it is known as Penicillium chrysogenum, but now previously it was known as notatum. Okay, so this produced antibiotics which is used for killing or retarding the growth of the of the microorganisms which cause diseases in our body. Okay, they are also used in synthesizing fats and proteins, and also they are for vitamins and enzymes. They're for synthesizing vitamins and enzymes. Okay, in industries, first in the breweries industry, that is for making wine. Okay, breweries industries, they use this fungi for fermentation of grapes, then barley maltose, okay, for beer done by yeast, which is known as Saccharomyces cerevisiae. Okay, cerevisiae is the yeast which is used for fermenting grapes and maltose for making beverages. Okay, in the brewery industries. So that ethyl alcohol and ethanol is produced. How this process works on? First, the grapes are crushed and then fermented in large vessels. Okay, first the grapes are crushed and then fermented in large vessels. Then the process is, this fermentation is carried out by adding yeast cells to the crushed grapes. Okay, which is either added or they are naturally present there. Then ethanol, which is produced, that is concentrated about 12%, which kills the yeast cell. Okay, if the concentration of ethanol which is produced is 12%, then it can kill the yeast cells. Now, the wine which is left after killing of the yeast cells, the wine which is left, that spirit, wine is what? It's a spirit which are whiskey and gin, having much higher alcohol content, and obtained, which is obtained by distilling of the fermented mixture. So after fermentation, ethyl alcohol is produced. When its concentration becomes 12%, yeast cells are killed. Now, when this after this ethyl alcohol, which contains 12% concentration, and after killing the yeast cells, the substance which is left, that is the wine. That is what? That is the spirit, which is whiskey and gin. Okay. And that has higher concentration of alcohol, and that is distilled from the fermented mixture, and that is known as wine. So spirit, which is whiskey and gin, and containing higher percentage of alcohol, and it is distilled, and the substance which is obtained, the fermented mixture which is obtained, that is known as wine. Okay. Now, the constituents of wine are variable. It contains amount of water, alcohol, certain amount of water is there, alcohol, carbon dioxide, sugar, and a few other substances. So the reaction proceeds in this way. Glucose, okay, the grapes which contains glucose, that anaerobically it is broken down by the yeast to ethanol, carbon dioxide, okay, ethanol and carbon dioxide. Okay, so this is the process how which wine is prepared, and the characteristics of wine is that how much pure the wine is, or how good is the wine is. We will be able to know by if the wine quickly absorbs into the body. If the wine is quickly absorbed into the body, then it's a very good wine. Where very mild quantities may be a stimulant, but large quantities can cause harm in many ways. Okay. If a person is taking, and if he takes in a mild quantity, then it acts as a stimulant to the body. But if he takes in large quantities, then it can cause harm to its body in many ways. Okay. Then alcoholics who drink too much, okay, those persons may develop body ailments like liver cirrhosis. It's a very common disease of the alcoholics. Okay, that is liver cirrhosis. Fatty liver, you can say. Okay. Then in baking industries for bread making, okay, that is also done by yeast, where they give out carbon dioxide, which makes the bread fluffy and spongy. Okay, so that is done by yeast. A mixture in this process, a mixture of flour, that is atta or maida, then some fat is used, and salt is given, then water is given, and a dough is made. Okay, and the starch of the flour, it gets converted into sugar. Okay, then the yeast is added to the dough, which increases its original volume, and when it is baked, when the dough is baked, after adding the yeast to it, okay, the gas will bubble out, and it will expand the dough, okay, and give the bread a light and spongy texture. It will light. If you have touched bread, no, you have seen that it becomes light and fluffy and spongy. So that is due to the carbon dioxide which has been released by the yeast. Okay. Then in the cheese making industry, it is used. In the cheese making industry, what is cheese? It's a valuable food which contains high quantities of fat and protein. Okay, cheese is a very valuable food which contains high quantities of fat and protein, along with calcium, phosphorus, vitamin A, and some quantities of vitamin B. Okay, so very high quantity of fat and protein, along with calcium, phosphorus, vitamin A, and vitamin B. Okay, that is a cheese. That is the cheese. Okay, it is prepared from what? It is prepared from milk. Okay, and the fungus which is used here is the lactobacillus fungus with the uh, milk fungus, you can say. So lactobacillus fungus is used. First, the lactobacillus fungus is used for curdling the milk. Then after the curd is prepared, that is separated from the whey. Whey is what? It is the liquid after after curdling of the milk, the liquid which separates out from the curd, that is the whey. So from that whey, curd is taken out, separated. Then moisture is removed from the curd, and that curd is known as cottage cheese. Okay. Now, further moisture is removed by adding salt to it. Okay, so that microorganisms, undesirable microorganisms, does not grow there. Now the curd is kept in a suitable temperature and humidity. Flavor of the cheese comes from the microorganisms which is added along with the lactobacillus in the first step. That is when the curdling was done, along with the lactobacillus, some other microorganisms are added to that milk so that the flavor comes to the cheese final product. Okay? So this is how the cheese is made in the cheese making industry, cheese processing. Okay. Then in the mushroom cultivation, mushroom is a very popular fungus. Okay, so mushroom are saprophytic multicellular fungi. Okay, they grow on dead and decaying leaves, wood logs, etc. Fruiting bodies of mushroom, they remain above the soil. Okay, the fruiting body, they grow on the dead decaying leaves, wood logs, etc. But the fruiting body, that remains above the soil. Edible mushrooms are agaricus bisporus. Okay, they are the important source of proteins and vitamins. These mushrooms provide us a large amount of protein and vitamins. A large number of them, they grow wild in nature. Most of them are mildly or severely poisonous. Maximum of them are mildly or severely poisonous. Okay, very few are edible mushrooms. Then the species which are cultivated in India are Agaricus bisporus. They are known as white button mushrooms. Then Volvarella, they are paddy straw mushrooms, and Pleurotus, that is oyster mushrooms. These three species are cultivated in India. They are cultivated generally indoors, and so very little land is required. They can be grown on substrate or compost, which is based on various agricultural waste, which in turn are recycled. So the agricultural wastes, okay, those are used as substrate or compost. These agricultural wastes are recycled and then they are used for substrate and compost. Okay, on which these mushrooms grow, and as they can be grown indoors, so land requirement is very less. Most popular variety which grows in India are Agaricus bisporus. Okay, so how the cultivation proceeds? First is the composting, where the mixture is prepared of wheat or paddy straws, then chicken manure, then some organic and inorganic fertilizers are used. Okay, so wheat and paddy straws are used, chicken manures are used, and some organic and inorganic fertilizers are used for the compost making. So they are used as compost in certain proportions. They are used, and the temperature that should be about 50 degrees centigrade around 1 week. Okay, then comes the next step, that is spawning. Here the mushroom seed is grown. Okay, the mushroom seed having mycelium of the selected type of the mushroom introduced in the compost. So what happens after this composting is done? Mushroom seed is introduced inside the compost and allowed to spread for a couple of days. So for composting, 1 week is required. Composting is done. Then mushroom seed is introduced into the compost and allowed to spread for a couple of days. Okay, so what is the mushroom seed? It is the mycelium of the selected type of mushroom. That is the mushroom seed. Okay. Then that is you allowed to spread for a couple of days. Then comes the casing. How does the casing is done? By spreading a thin layer of soil over the compost. So compost is there, my mushroom seed introduced, then a soil layer is covered over the compost. So that is casing. Okay. What does casing give us? That gives support to the mushroom, and it provides humidity. It prevents quick drying of the compost and helps to regulate the temperature. Four things that is done by the casing. Okay. So one is that it supports the mushroom. It provides humidity to the mushroom. It prevents quick drying of the compost, and it regulates the temperature of the soil. I mean the compost. Okay. Then, but for the arrangements, for this casing arrangement, there must be this casing must be made of air circulation around the beds. Okay, for this arrangement, for this casing, there should be air circulation around the beds. Okay, and temperature must be kept low, 20 to 25, so that the pests and other diseases does not grow. That growth of the pests and diseases are avoided. Okay, so the temperature should be kept low and air circulation must be kept around the beds. Okay, this should be there for the casing. Then comes the step of excuse me, cropping and harvesting. Then this occurs in three main stages. Okay, the mycelium grows within 2 to 6 weeks. So this is done. Casing is done. First, composting is done. Then yes, spawning is done. Then casing is done. This is kept for 2 to 6 weeks when the mycelium grows. And then what happens? Cropping and harvesting is done. Then tiny pinheads come out. No? Then these button stage growing bigger attain. When the button stage grows bigger, then marketable size when it becomes the full grown, when they are full grown, they are taken out. Okay, so 2 to 6 weeks they are kept as it is. Then when the pinheads come out and they grow to a marketable size, full growth it becomes, then they are taken out. Now, how to preserve them? Because mushrooms are very highly perishable, so their shelf life has to be increased. How that is done? Vacuum cooling is done. Okay, they are given gamma radiations and stored at 15 degrees centigrade so that pests and diseases does not grow there. Okay, so low temperature has to be done, vacuum cooling is to be done, freeze drying has to be done. That is sublimation of ice from frozen mushrooms at reduced pressure. Pressure is reduced and mushrooms should be frozen. Sublimation of ice. From the sublimation of ice. Mushrooms should be frozen for sublimation of ice and reduced pressure. So that is known as freeze drying in a solution of citric acid, ascorbic acid or drying. Okay, that is done so that they can be preserved. Okay. Now, what is the nutritive value of the mushrooms? They are the excellent source of vitamins and minerals. Okay, they give niacin, pantothenic acid, and biotin. Okay, during cooking, canning, and dehydration, its vitamins are retained. That is not lost during cooking, canning, and dehydration. Okay. Okay. Now they contain 85 to 95% moisture, 3% proteins, 4% carbohydrates, 0.3 to 0.4% fat, and 1% of minerals and vitamins. Okay, main thing they have moisture, 1% of minerals and vitamins, 3% of proteins, 4% of carbohydrates, and fat is very less, that is 0.3 to 0.4%. So this is the end of this chapter, economic importance of bacteria and fungi. Okay, next chapter will be again of biology, nutrition chapter, I think so. So that I will start with. And before that, I will request you, if you are liking the video, press the like button, share with your friends. If you have not subscribed yet, or you are a new viewer to my channel, please do subscribe so that when a new video is uploaded, it will notify you. Okay. And please do meditate before you start your day. Do maintain a chart so that will help you to track your studies. Okay. Chart of your studies. Okay. A routine, make a routine of the subjects which you want to study the whole day, and make it in the morning, and then at night you can check how much you have completed. That will keep you updated and well versed before your exams. Okay. So please do stay tuned with me for the next video. And if you have any doubt, please do write in the comment box. You can write also in the email ID, that is kasturi74@gmail.com. Okay. So thank you for joining, and bye till then.